Good morning and welcome to today's MPG Primer. We're very excited to have as our speaker today, Dr. Alyssa Lawler, who's a postdoctoral fellow at the Stanley Center. During her graduate work at Carnegie Mellon University, Dr. Lawler used a combination of experimental and computational approaches to identify DNA sequence features that confer cell type-specific gene activation. She used these features to design probes that enable labeling, isolation, and manipulation of neuron subtypes for research and gene therapy. And in her ongoing work here at Broad, Alyssa is studying schizophrenia risk genes and their knockouts in mouse models in the McCosco lab. So again, thanks for being here. And Alyssa, we're really excited to hear about your work. A reminder to the audience that if you have any questions, we have microphones. So we encourage you to raise questions at any point. And if you're joining us remotely, please post your questions in the Q&A and we will voice those in the room here as soon as we can. Thanks, Alyssa, and please get started when you like.
Okay, thank you so much for the introduction. I'm really excited to be here with you all today. Excited to get to share some of our work on Tacit from the end of my PhD with my new home here for my postdoc. The work that I'm going to present today is on Tacit, a tissue-aware conservation inference toolkit for relating enhancer genetic variation to complex phenotypes. And this comes at an opportune time. This work is available currently as a preprint, and the final version is in press and will be available very soon. So do keep an eye out for that. Okay, so before I get started to acknowledge the Fenning Lab at Carnegie Mellon University, where I did my PhD and where this work was completed. And I also wanted to acknowledge my two co-lead authors on this work, Irene Kaplow, who's finishing up her postdoctoral fellowship, and Daniel Schaefer, who is actually a really extraordinary undergraduate researcher who worked on this project with us. And he's now about to start his PhD journey. This work is also part of the Zoonomia Consortium and would not have been possible without the resources and community and support of Zoonomia. And you can learn more about that at zoonomiaproject.org. So phenotypic diversity across vertebrates is thought to have arisen from, yes, changes in genes, but also importantly, from changes in how genes are expressed. A species' ability to perform vocal learning, for example, is associated with specific patterns of gene expression in specific brain regions and circuits across songbirds and humans. In another example, there are tissue-specific gene signatures whose expression in a given species is tightly correlated with that species' lifespan and life history strategy. And that at least some of the genetic differences associated with the evolution of complex phenotypes are likely to function in enhancers, which help regulate the expression of genes throughout time and space. Genetic variation in an enhancer could, for example, change its affinity for certain transcription factors or other interactors, and thereby change its enhancer activity and potentially expression of the target genes. Enhancers are particularly well poised to potentially be a really important substrate for molecular evolution. There are many enhancers throughout the genome. We're talking millions of candidate enhancers in the human genome, regulating just tens of thousands of genes. Enriched for evolutionary conservation, but they tend to be much less constrained than promoters or genes themselves. And this is in part because enhancers can have very highly tissue or cell type specific function. So one salient example of how enhancer sequence changes can contribute to complex phenotype evolution comes from a really nice study from 2016 about the evolution of limblessness in snakes. This can be traced back to a single enhancer called the ZRS enhancer, which regulates the limb-specific expression of the sonic hedgehog gene during a specific time in development. And This DRS enhancer is thought to have degraded over time in the snake evolution, and that this contributed to loss of limb in snakes. And if you put the sequence from a cobra DRS enhancer and switch out the endogenous mouse DRS enhancer for this cobra sequence, you can show the functional consequences, and the mouse fails to develop limbs and becomes serpentized. So this is a one really great example of enhancer phenotype evolution. But examples are actually quite far and few between at this stage because they can be really tricky to identify. Luckily, fortunately, a lot of recent advances have sort of coalesced to make this a great time to innovate in this space toward facilitating identification of the relationships between enhancer activity and phenotype evolution. For one, genome sequencing projects like the Xenomia project and others have now provided high quality genome sequences for hundreds of species. This is a very new type of resource. Accompanying this, there's been great advancements on alignment tools, for example, reference-free multi-species whole genome alignments like the cactus algorithms, and these allow us to map the coordinates of non-coding regions across species. Finally, there have been advances in phylogeny-aware statistical methods for phenotype association, including phylum packages and phylogenetic permutations, which I'll talk more about later. We don't have the enhancer data. That's the only thing we're missing right now. And it's not feasible to collect enhancer activity data from hundreds of diverse species and many tissues. Our solution is to impute the enhancer activity from genome sequence with machine learning. So we can take the enhancer activity data that we do have from a small number of species in a specific tissue, and we can use this to learn the rules that govern enhancer activity in that tissue from sequence, and then predict in all these new available genomes at the orthologous enhancer region whether those sequences are likely to be active. Why machine learning and not something simpler? This is a great question we should always ask ourselves before jumping into the latest and greatest technologies. I'll illustrate why with some information from a great paper by Wang et al. In 2020. What they did is they took enhancer orthologs across mouse, human, zebrafish, and even all the way out to sea sponge, spanning, I think, 100 million years of animal evolution. And they took each of these sequences into a reporter assay in zebrafish, and they found, kind of surprisingly, that despite very significant nucleotide divergence, many of these enhancers still drove very similar cell type specific enhancer activity in the zebrafish. They attributed this to a deeply conserved cell type specific regulatory code across animals that is permissive of flexible motif configuration. Because when they looked a little bit closer at these precise sequences, yeah, although the nucleotide sequence is very, very different, there are conserved transcription factor binding site motifs that are just in different arrangements in these sequences. So this is really great news because it suggests that there is a learnable code that we could apply across animals. It's just a really complex code. So we can't rely on simple nucleotide divergence mechanisms to infer activity change. And we also need to take into account that these codes are cell type specific. So we turn to convolutional neural networks, or CNN, which achieve state-of-the-art performance on relating enhancer sequence to enhancer activity. And the idea is that while enhancer sequence across species may evolve rapidly, these complex rules that determine enhancer activity from sequence stay consistent. And so if we learn those rules once, we can apply it to new sequences and new species to infer activity. Brings us to the framework for our toolkit. called Tacit. I'll go into each of these components, but briefly, we're first going to talk about training sequence-based machine learning models to predict enhancer activity from sequence in specific tissues. In the talk today, I'm going to be focusing on our model for motor cortex open chromatin data. Then we use these models to predict across 222 mammalian genomes from the zoonomia to predict the enhancer activity at the orthologs. And then we will use these predictions toward identifying enhancer phenotype associations in a phylogeny-aware approach. So in those three parts, we'll step through Tacit. And along the way, I'll be describing the methodology and innovation, showing results from our motor cortex associations, including associations with brain size and social strategy. And I'll also be highlighting the new resources provided and how you can take advantage of Tacit for your own work. To that, I wanted to mention this is a primarily evolutionary biology driven piece of work, but I know many of you are motivated and interested in human disease. So I just wanted to explicitly mention some of the interplay here and how you could think about this. I'm a big believer that disease genetics could be informed by trait evolution genetics and the interpretation of these, and vice versa. For example, the species phenotype association that we're testing can be disease related directly or indirectly. I mentioned we'll be looking at enhancers associated with brain size. You could speculate that human genetic variation in these regions could also potentially relate to diseases involving micro and macrocephaly. Enhancers within a specific disease deletion site, the deletion locus for the Williams-Bjorn syndrome. And I won't be able to talk much about this today, unfortunately, but wanted to mention some really exciting extensions going on the disease side of Tacit. This is work from the Fenning lab led by Bedoy Fan, and he's found that the predicted enhancer activities from Tacit provide a unique conservation metric that is really useful actually to help improve fine mapping of human disease associated genetic variants. Okay, so getting into the models, we'll first talk about training the tissue-specific enhancer model. This is a basic overview schematic of a convolutional neural network classifier architecture. It contains an input sequence layer, which is one hot encoded, and then a series of convolutional and max pooling layers to scan the sequences for subsequent patterns and downsample. These go into fully connected layers and eventually into a sigmoid output, which represents the probability from 0 to 1 of enhancer activity. So these model weights within the model are learned through automated epochs of training, optimizing the loss. And then we manually iterate multiple versions of these models, tuning parameters, tuning architectures, until we get a model that's reliably able to accurately predict enhancer activity from known sequences. And these are transferable from training data to held out test data that the model hadn't seen before. Motor cortex models, for our evolutionary models in general, is really important for us to train two separate types of models for different purposes. For the motor cortex, have enhancer data available for 4 species: mouse, rat, macaque, and bat. So what we want to do is first train a single species model. So we're going to train a model using only the enhancer data information from mouse, for example. And then we're going to evaluate whether that model is capable of correctly predicting known enhancer activities from orthologs across the other species which we have data for. And this helps us understand whether the model with this regulatory code is generalizable across species. Once we've checked that off, we can then make a new multi-species model. So this is going to leverage all of the information that we have available to us from all species, and we'll be actually using this one for the prediction across the broader set of 222 genomes and imputing phenotype association. This is the actual data. What do the positive and negative sets look like? The positive set examples will be sequences underlying motor cortex open chromatin peaks for this model, and specifically ataxic data. We've further restricted these to non-exonic, non-promoter open chromatin region to increase the likelihood of enhancer function specifically. You'll hear me refer to these as OCRs, and I'm basically using open chromatin as a proxy for enhancer activity throughout this talk, and closed chromatin for inactivity. That is by no means a perfect equivalence, but the data works really well for this type of modeling, especially because it's a little bit more widely available, and ataxic in particular provides a really precise positioning in the genome to point us toward a small region of the affected effector sequence. For negative set examples, one type of negative sequence that our models get is always sequences underlying non-OCR orthologs of OCRs in other species, because we want this yellow highlighted sequence to be predicted as an inactive. We sometimes also incorporate sequences underlying non-OCRs that are OCRs in other tissues and cell types to help us get to the tissue specificity. This we found is particularly important to include in our cell type level models. And then if we need additional training examples, we can also include other regions of the genome that do not overlap in OCR, and these will be GC matched to the positive set. Here are the performance results from our mouse-only motor cortex model. I'll be showing a series of bar plots with the standard classifier performance metrics, area under the receiver operator curve, AUC, AUNPV, and AUPRC. And each of these metrics has a maximum value of 1 for a perfectly accurate model, so the higher the better. You can see the model is performing quite well at accurately predicting the enhancer activities of the test set that reflect the training set. Importantly and excitingly, if we evaluate this model on known sequence examples from the other species for which we have the data, we find that it can also accurately predict the enhancer activities in other species almost just as well as it can in mouse, even though it never saw any other species during training. And we can further subset these types of sets to clade-specific and species-specific OCRs. We can do really well across all of those cases. These are cases that are easily missed in previous enhancer models, but are going to be really important in our case for evolutionary association. Here are the equivalent performance metrics now for the multi-species motor cortex model. So we're now leveraging all of that data that we have from the 4 species, and this model performs really well across all the evaluation categories too. Additional tests we did to look at the generalization of these models across species. First, we looked at the mouse enhancer OCRs, and we found that the mouse positive examples, the further you move away from mouse in evolutionary distance, the population of mouse OCRs gets lower and lower predicted on the convolutional neural network. That's what you would expect through evolutionary divergence. We were also curious to see the most important things that the model was weighting during this learning task. Basically, is it learning a different set of rules for each of these different species, or is it a common set of rules? So we looked at the first principal component output of the first fully connected layer, and we found that it was separating by positive and negative populations, not species. So this is also good for generalizability to new species. These CNN models are available for you to use on our supplementary website. You can navigate to this link, which is in the paper, and click on the CNNs, and you'll find directories for our main models, including brain models, which includes the motor cortex model I'm describing right now, but also multi-species models for a specific subtype of neuron, PV neurons, and retina. And we also have a liver model that you can take advantage of. In addition to these, there are a number of other supplementary models. And so each of these will contain the files for the model architectures and weights. So you could load these into Keras, for example, and use the models to predict enhancer activity on your own sequences. The model definitions can get a little bit tricky, and there's a lot of details. So we also included a model encyclopedia where you can quickly reference the information for any given model. Great. Moving on to the next section, we get to do the fun part, take these models that we built and rigorously evaluated, and now apply them toward predicting OCR ortholog activity across 222 mammals. Here is a snapshot overview of motor cortex open chromatin activity predictions across the 222 genomes, which are along the x-axis in order of this phylogeny. And the enhancers we've also clustered by row, and you can start to see some enhancer patterns pop out related to common enhancers, clade-specific enhancers, and then those with more nuanced relationships as well. This is just a random subsampling of 1,000 enhancers on this plot. So the actual prediction tables that we've generated are about 100 times larger than what I'm showing here. And we predicted nearly 100,000 OCR orthologs across 222 mammalian genomes for the motor cortex. And we have separate additional prediction tables for the PV models and liver models too. Are also available to you in the form of prediction browser tracks. You can get these on the supplemental website, or soon they will also be available directly on the UTSC genome browser, where you can navigate to the Xenomia track hubs, and you'll be able to browse the genomes from the species in Xenomia. So I've just clicked for fun on the gorilla genome, and if you toggle the expression and regulation section for Tacit open chromatin, you can display our tracks onto the gorilla genome, and this will be a normal genome browser that you can click and drag and zoom in and out or navigate to wherever you want. In this screenshot, I've just navigated to a locus of the gorilla genome near the SLC25A24 gene, and you can see some of our enhancer orthologs up there where they map to, and the color is going to correspond to their predicted activity out of the CNN. If you click on a specific specific peak, you can get more information about that peak, like its exact positioning, and then the signal value, which corresponds to the CNN output, which is that 0 to 1 probability of enhancer activity. Next, we want to take these predictions and perform the OCR phenotype associations to look for OCRs whose sequence is associated with the evolution of a complex trait. In order to do this, we are basically implementing a version of phylogenetic least squares regression to ask the question, does the trait have an association with the predicted enhancer activity that cannot be explained by phylogeny alone? And that last part's really important. To illustrate what this means in cartoons, take this toy example of a evolutionary tree with a binary phenotype, or yellow or blue. This phenotype would actually not be a good candidate to test in this case, because the phenotype is completely confounded by these two major clades in the phylogeny. So any enhancer that's associated with this phenotype, you don't know whether it's actually associated with that phenotype or with any number of phenotypes that are clade specific between these two different clades. So this wouldn't be an association for us. Take another toy example where the phenotype is more distributed throughout the tree, and we would want to look at our predicted enhancer activity for a given enhancer. Note in this case that a regular regression model might call this a significant association, because most of the yellow species have high predicted activity and most of the blue species have low predicted activity. But again, when you take into account those phylogenetic relationships, you can see that the tree topology is actually a better explainer of the enhancer activity than the phenotype itself. So this again would not be a good association. So instead, what we're looking for is to identify relationships such as this, where there's convergent association between enhancer activity and phenotype activity that's not explained or confounded by the tree itself. Implementing two existing methods for binary trait and continuous trait association. We also made modifications to these methods. Daniel made code for the binary association that can reduce the runtime up to 10-fold. So this allows us to really scale these up to tens of thousands of enhancers for the first time. Next thing we do after the initial association is we end up computing an empirical p-value for these. Use permutations. Permutations are a type of permutation that account for trait topology in the phylogenetic tree. Again, very important. So essentially what these end up doing is shuffling the phenotypes around the evolutionary tree. And then you would test for association between the shuffled phenotype in your enhancer to create a background distribution. And what you're going to calculate is going to be the percent of trials that have a better association than your actual observed enhancer phenotype pairing. So if your enhancer phenotype pairing ranks second out of 1,000 permutations when you shuffle the tree, that would give you a value of 0.002. We tend to do this over 1 million permutations per enhancer per phenotype, and we end up doing multiple hypothesis correction on these at the end as well. Wanted to mention we've also implemented a staged permutation strategy, which can also minimize the runtime and help us scale up. Okay, so getting into some results of some of our motor cortex trait associations. The first trait that I'm going to talk about is a continuous trait, and this is brain size, specifically brain size residual when regressed for body size. There is a consistent and particular relationship between a species' brain and body size that follows this formula. And some species fall right along this line and have the exact predicted relationship between brain and body size, like the mouse. But some species, like the human, have a particularly large brain for their body, or vice versa. So we're going to be looking at this measurement, that residual, as a continuous phenotype to look for associations between that and enhancer activity across our predicted enhancers. We found 49 motor cortex OCRs that were associated with this brain size residual. A few pieces of evidence for why we think these are plausible biological associations are that 42 of these enhancers were nearby brain development or brain tumor growth genes. Additionally, 22 have a known physical interaction with regions in the genome near that gene too, through high-C data in mouse or human cortex. So this lends some credence to the fact that these could be functioning as enhancers. We also see specifically 14 genes that are nearby known genes that are related to micro and macrocephaly in people. And 5 of those overlap the high-C genes too. So we think this is really uncovering new biology, and it's important to mention that none of the phenotype OCR associations were able to be identified with existing methods that rely on nucleotide conservation and are not tissue aware, highlighting the added knowledge from Tacit. Zoom in on one of those OCRs, which is near the LRIG1 gene. You can see that the association between the brain size residual phenotype and the enhancer predicted activity are particularly strong within the cetaceans clade and the primates clade. So it's 250 kilobases from this LRIG1 gene. LRIG1 is known to slow and delay the differentiation of neural stem cells through interactions with the EGFR pathways. And this is one where there is a documented 3D interaction between this OCR region, or the region of the genome containing this OCR, and the region of the genome containing the LRIG1 gene. So we could speculate that it could be acting through enhancer activity evolution related to regulating LRIG1. That's just a prediction. We haven't tested that though. Moving on, the last trait that I wanted to talk about is a binary trait for social strategy. Animals can fall into different social strategies, including solitary living or group living, for example. It's thought that the solitary state is ancestral for mammals, and that heterogeneous group living strategies have evolved multiple times throughout multiple clades in mammalian history. So this is a good phenotype candidate for us to test. In this case, we took a targeted OCR phenotype association approach within a specific locus of the genome. And if you have these sort of hypotheses, this can help with the burden of multiple hypothesis correction if you take the targeted approach. So in this case, we decided to look at solitary associations within the region of the genome related to Williams-Bjorn syndrome. So patients with a core 1.5 megabase deletion in this area of the genome have a number of cognitive and social differences. And one of those is they're characterized by extreme hypersociability. So we were interested in relationships between this and social evolution. In particular, 2 genes, GTF2-IRD1 and GTF2I, have also been associated with cognitive and social changes in mice, suggesting this might play a role in animals too. And if you knock down GTF2I in particular, this increases nose-to-nose sniffing in mice, so maybe hypersociability there as well. So we decided to look at, are there motor cortex enhancer candidates in this region that are associated with the evolution of social strategy in mammals? We did find one such OCR for the motor cortex in this region, and it was within this region right near those 2 GTF genes. This is what the association looks like, and you can see that social species tend to have higher predicted activity at this OCR from our models, while the solitary species tend to have lower predicted activity across clades. So I think this is a really cool little window into how we can think about phenotypes and disease interactions, and how we can learn from interpretation of both together. The tools that we use for phenotype association and the modifications that we've made are available on our GitHub. This will also be linked in the paper, and you can look at the README for details about some of the key functions and utilities that you can use. So lastly, I'll just go quickly over a flowchart of how you could implement these resources. The things you need to start with, you need two things. You need your table of species phenotype assignments. That can be your binary or continuous phenotype associated with each species name. And then you need your table of species by OCR predicted activities. So that can be ours or ones that you generate with Tacit models. It could even be something other than predicted enhancer activities if you want to use these tools for other associations. And what you're going to do is take these two inputs and run either the continuous or binary trait association pipeline. You're going to run it twice. You're going to run it once without permulation to get the actual association of your observed enhancer, and then you're going to run the permulation. So shuffle the tree and test for association many times. And this will result in two output tables, one with your raw enhancer associations, one row per PCR, including information for that association and the coefficient. And then you'll get many long tables where each row is a different permulation. So each OCR has many rows because it has many permulations. And then to consolidate the information from these outputs, you'll use one of our functions to compute the permulation p-values. And this will do the empirical p-value calculation. And then you'll also want to correct these for multiple hypothesis testing too. Okay, so that brings me to the end of what I wanted to share with you today. To recap some of the key innovations, we've begun to leverage these cutting edge methods and resources in an innovative way to start to interpret genetic variation in non-coding regions toward phenotype evolution. We've been able to scale the CNN enhancer predictions and associations to tens of thousands of regions across hundreds of species. We've also described speedups for the methods along the way, including the trait association statistical method. To recap some of the results I've shown, we have these motor cortex OCRs that are associated with brain size, including the one I highlighted near LRIG1. We also point out that one group living associated motor cortex OCR within the Williams-Barron syndrome deletion locus. And then our new resources provided include the models themselves, the prediction browser tracks, and the code for Tacit phenotype association implementation. A couple notes on future applications and extensions of Tacit. We are hoping that people with specific interest in their own tissues and things will want to extend these types of methods to new tissues and cell types and new phenotypes. If you're doing that, just some recommendations to get started. It basically takes at least high quality tissue specific enhancer data from at least 2 species. So you can do the proper evaluations. You need to properly evaluate those models on subsets of species and clade specific enhancers. And then overall, you're going to want to choose a phenotype that is not confounded by phylogeny and match that to an appropriate tissue for testing. I mentioned at the beginning of the talk, we're also working on applying this toward interpretation of disease for human genetic variation, and that's some really exciting ongoing work. In addition, we are working on developing methods for experimentally testing and validating enhancer ortholog activity in vivo. There's not really a good high throughput method that can do this in a tissue specific method in vivo. So we've been working on developing an in vivo MPRA in mice a massively parallel reporter assay to scale up this testing. And that work is also in development, but it's available on bioRxiv. And with that, I want to acknowledge again the Fenning Lab at Carnegie Mellon University. That's where I did this work and my PhD, especially the PI Andreas Fenning and Irene and Daniel. I also want to thank the Zoonomia Consortium, especially Eleanor and Sharfson for the organizers, various funding sources that contributed to this work. And if you want to keep up with these projects and publications, you can visit the lab website at fenninglab.org or zoonomiaproject.org for the broader Zoonomia Consortium. And this is my contact if you want to get in touch now that I'm here at the Broad with you all. So thanks for listening and happy to take any questions.
I wonder, have you looked at those filters in your CNN model, what they are? And are they kind of, what after your training, are they also get those filters that some are well-known sequence for enhancer?
Yeah, that's a great question. Yeah, so CNNs are a little bit elusive for interpretation, but we have done some attempts at model interpretation to understand the subsequence features that it is learning. I don't have that with me today, but there's some information in the paper. So we've extracted highly weighted subsequences using TF Medisco, and we did find some known transcription factor binding motifs that make sense for the relevant tissues, as well as novel tissues or things that might look like combined motifs or other types of the regulatory code. But yeah, we've started to look into those more, and we would like to be able to understand then when we propagate this genetic variation, what exactly parts of potentially known transcription factor binding sites are getting changed in such a way that affects the activity. Yeah.
One more is, if I'm interested in a certain region, and I know that's enhancer, does your model can help me identify which part of sequence of that enhancer maybe has a higher contribution to classify as a enhancer. So
yeah,
I can pull out the sequence, say, oh, that's maybe another conserved sequence, and maybe it's just a tiny part of the huge long enhancer make larger contribution rather than the whole sequence of that part of it, of that enhancer.
Yeah, that is often a scenario of something that we want to look at for a particular enhancer. Yeah, so you could similarly do that by attributing through tools like TF Medisco or something like that. You can attribute importance scores to every nucleotide within your enhancer region using the outputs from the CNN models. And you could also look at those across the species orthologs to try to identify where that conservation is showing you, which might point you toward the particularly important subsequence features.
Thank you.
Yeah, thank you.
Thank you for an excellent talk. I have one other question, which is I can imagine ATAC is ideal training because it's fairly dense. You get a lot of regions to train on.
Yeah.
Have you thought about subsetting like ATAC that overlaps certain histone markers like H3K27 acetyl to look for active enhancers?
Yeah, I think that's a really great direction to improve in the future. Yeah, incorporating histone marks or other markers to improve the definition of our enhancer activity. Yeah, I think the challenge is getting those types of data from the multiple species and from specific tissues and cell types, because those Tacit, you don't need as many cells, so you can do it on smaller tissues and cell types. But yeah, where available, we would love to incorporate that into the definitions to improve the models too.
Great. Well, thank you again for an excellent talk.